1 scoop of vanilla ice cream. Thank you. Sir? Hey, sir?
Yeah.
That'll be $5.
Oh, I forgot. It's so good.
Courtney. Vanilla Kano.
That's really cute, Brad. I hope your fingers are clean.
Yoda! This is human ice cream, not dog ice cream. Ugh, gross. I was gonna eat that.
Quit whining. His mouth is way cleaner than your hands are.
Yeah, right. I've seen him when he tries to clean himself.
It's my little Jedi.
I don't know how someone arrived at the brilliant conclusion a dog's mouth is cleaner than a human's. Seriously, a dog's tongue is his toilet paper. Draw a conclusion from that. But if that were the case, wouldn't every pet owner get sick?
Ever since I was little, the natural world has always fascinated me. Exciting discoveries and mind-blowing things are revealed every day. And I'm learning there's a lot more to do than sitting in a cubicle or wearing a pocket protector. My mission? To explore and journalize real careers in science, technology, engineering, and math. These are the STEM journals.
I'll admit, when I was a kid, I used to fake being sick all the time. I mean, it was awesome. I got to lay in bed and watch TV and play video games and miss school. And Mom would bring me whatever I wanted. It was awesome. Until I actually got sick one time and realized how miserable it is. I mean, laying in bed, hacking up a lung, coughing, and I had so much schoolwork to make up. Why anybody would willingly hunt diseases is beyond me. But now with my crazy quest to learn more about science, here I am in the middle of nowhere with wildlife specialist Ann Justice Allen, who's gonna talk to me about diseases within the fox population. What specific diseases are you looking for within foxes?
Foxes get parvo and distemper just like domestic dogs. They also carry rabies, which is a disease that people can get. And so we look at that as well.
What about the foxtrot? Heard that's pretty contagious, huh? You know, like...
Oh, Brad, you okay?
What was that?
Well, that's a foxhole.
Well, that's convenient. So what I was gonna ask before you rudely interrupted me by planting this foxhole, the diseases that you find within the foxes, how does that pertain to humans?
We're looking at the inadvertent or unintended consequences of human activity on wildlife populations, such as our domestic pets carrying diseases out to our wildlife and affecting their ability to reproduce and maintain their populations. Let's get going. Got traps to check. We set these traps last night to try and catch foxes during the night for this morning. Here's our first trap.
Where's the fox?
Well, sometimes wildlife biology is a little bit unpredictable, and we don't always catch them when we expect to.
How do you lure them in there?
We do use food sometimes. Interestingly enough, they are attracted to obsession, the perfume, and that's sometimes used to get them to come to these traps.
So foxes even love Calvin Klein?
Yep.
That guy just knows what he's doing.
It's amazing.
Oh my gosh. Didn't you say you're a veterinarian?
Yes, that's right.
I thought a veterinarian was like, oh man, please fix my sick kitty.
Well, we do that too, but we also work on wildlife and in research institutions, food safety, just about anything you can think of.
Okay, we better find a fox today and trap one, because if not, it's gonna be super boring.
I'm fully expecting that we'll get at least one.
Ooh, I might be able to help us out. I'm working on my fox call.
That's all right. Allie and Carrington already have one over here.
Told you my fox call worked.
No, Brad, that poor little guy's probably been waiting there since sometime in the middle of the night.
Okay, agree to disagree, but we got a fox now, we didn't before, so
All
you're
right.
welcome.
Well, we're gonna move this guy out from underneath the bushes here, get it into this bag.
What are we trying to achieve by taking him out?
We're gonna get some blood and we're gonna get a poop sample and have those tests run for the diseases that we talked about.
You don't put him to sleep through this? He just kind of stays awake and hangs out?
No, these guys are pretty docile.
I'm telling you right now, that thing is way too cute to have parvo. I don't even think we need to test him.
Well, that's the thing, is you can't always tell that they've got something until you've done the test. You know, sometimes these guys have fleas, and sometimes fleas carry disease like plague.
Huh.
Okay, now for the poop sample.
I understand the blood, but why do we need the poop sample?
Parvovirus is an intestinal virus, so the virus ends up in the poop, and we will use a swab to collect a sample of that poop, and then use this test kit to detect the virus. Easy little guy.
I'm gonna let you do this one, okay?
Well, maybe you'll get a chance here yet. Get a little sample. You're a stinky boy. Yeah, we're done.
Man, they are fast. If you just did that to me, I'd probably be running that fast too.
Looks like you've got a job after all, Brad.
Oh man, poop cleaner?
That's exactly right. More is better.
There's this. Sure you don't want this?
No.
Okay.
Bag, please. Thank you.
When I return to civilization, it's time to mix up some mouth-watering fecal floats. More like vomit-inducing fecal floats.
I'll have thousands of students.
I'll have a lot of teachers.
Many of these bright young minds will change the way we live in the future.
Maybe I'll be the one who finds a cure.
With STEM education, science, technology, engineering, and math,
I'll have a great career. And maybe I'll change the world.
no doubt.
Copper Point is proud to sponsor STEM education because students who excel in science, technology, engineering, and math will solve the challenges of tomorrow. Copper Point.
Okay, so we got our poop and our blood. Now what do we do with it?
Well, we're gonna go ahead and run a parvo test. We're gonna take some of the poop on a swab, mix it with the blue liquid that's at the tip of the swab, and then drop it on this container, and it'll tell us whether or not these animals have parvovirus in their system right now.
Can't wait. What's the big deal with parvo anyway?
We're going to compare these animals out at Palo Verde with the animals near Mesa. The hypothesis is that the animals near Mesa are more likely to be infected with parvo from the pets that live in town there. We're going to test that hypothesis here. As soon as this liquid moves up, then we'll push it down, wait for about a minute, and it should tell us whether or not we've got parvo in their systems.
It's exciting.
When the results come out, then that can be exciting. That's the snap part of it.
And snap test.
Exactly. All right, why don't you put like 4 drops right there in the middle of that. Just squeeze that bulb right there.
Me?
Just don't try and aim from too far away. You want to get pretty close and just let it drop straight down.
How many drops?
4 drops.
1, 2, 3, 4. Oh, that was 5. Is
That's
that
all
gonna ruin
right.
our test? Is it
No.
gonna double parvo?
No, not at all. Okay.
Now we have to wait.
Wait, and see, this one's already turning blue. These tests come with an automatic internal control system to let you know that the test is working properly. So that's what this first blue dot is right here. If the animal has parvo, then there'll be another blue dot up here.
Is there any other tests that you do?
We're going to test for antibodies in their blood, and we're going to send the blood sample into the lab. But the other thing that we can do is we can do a fecal float.
A fecal float?
A fecal float.
Sounds delicious.
What we do is we take this fecal sample and we put it in a salt solution, and if there's any parasite eggs in there, they'll float to the top of the salt solution. But we need to get a swab out of one of the containers over there.
Okay.
And if you'll pass me that liquid that's on the counter over there, we'll put some in this test tube. There you go. Get some of the poop on there.
Can I just
Yeah,
grab it?
you could grab it. You've got gloves on. Stick it in the tube.
Okay.
Okay. Now you gotta mix it all up. Nice and mixed up. Okay.
Looks
that's
good. It smells good.
Stick it back in the rack there, and we're gonna put a cover slip on it, and it needs to sit for a few minutes until it's ready. Got to give those eggs a chance to float to the top. So that one sits there for about 15 minutes.
So can we tell visually, or do we have to?
That's gonna have to go under the microscope. We only have a single blue dot on each one of them, so none of these animals actually has parvovirus in their system right now. We'll take our fecal float that we've, it's been sitting here for a little bit, and put it on a slide, and we'll look at it under the microscope.
Just because we didn't find any parvo today from the foxes, that means they've never had it, or that just means they don't have it right now?
They don't have it right now. We're going to send the blood in, and the lab will look for antibodies, and they'll tell us whether or not the foxes were exposed to parvo in the past.
What is this here?
This is the fecal float. We're looking for the eggs of intestinal parasites. There's a parasite egg right there.
This one?
Yeah, the thing with the 2 eggs looks like a double-yolked egg there almost.
Finding this doesn't necessarily mean that it's harmful.
No, the foxes can live with this quite easily.
You mentioned some of these guys can carry fleas, which also can carry plague. Didn't that take out like half of existence a few years ago?
We do sometimes see cases here in Arizona, not in people, but in prairie dogs. You should go see the folks up in Flagstaff at TGen North. They've got a research project on prairie dogs right now.
Dave! Hey, it's Brad. Hey, man, sorry I'm late. I had to come in from Phoenix, and there was so much traffic.
Oh, no worries, Brad, but we do got to roll.
Where are we going?
We had a call from the county health department. There might be an outbreak on the other side of town.
An outbreak of what?
The plague.
The plague?
We think some prairie dogs on the other side of town may have died of the plague. It could be that it's just so cold out they're not moving around, but they may have all died off from the plague.
Okay, the plague like Black Death, the plague?
Yeah.
Okay, how about this? Wouldn't a place with the Black Death be a perfect place not to be?
Coming up next in my STEM journal, when Anne brought up the plague, I thought I was in for a history lesson. Now I'm looking for the real Black Death right here in Arizona.
Jack is a healthy, happy first grader. Loves Legos, loves baseball. As a parent, when your child is diagnosed with cancer and you think you're going to lose your child, you do not know what to do. The only place that I felt comfortable taking him was Phoenix Children's Hospital. The fear that first impacts you is fully taken away. Their sole goal is to make your son better. Jack is cancer-free because of the physicians at Phoenix Children's.
STEM journals, supplemental. The plague wiped out something close to a couple 100 million people during the mid-1300s. Infected people were burned to ashes in a frantic effort to stop the disease from spreading. And like how the nursery rhyme Ring Around the Rosie ends, we all fall down. That almost happened to humanity.
Let me get this straight. The thing that we're going to hunt right now is the same thing that killed off like half of the human population a few hundred years ago.
That's right. You know, the Black Plague, a lot of people don't realize, is the very same disease that we call plague in our backyard here in Flagstaff.
See, which confuses me because the Black Death epidemic happened in Europe, right? So how did we have the privilege of having it here, and how did it get here?
The plague has really spread around the world. In fact, it didn't come to the New World until just about 100 years ago when it came over on sailing ships from the Far East. The rats and their fleas got off the ships and then got into the local wildlife populations and started infecting our rodents here in North America.
So that's why we're hunting prairie dogs today.
That's right.
I got it. I got it.
Oh, Brad,
Yeah. Yeah.
come on back here. What we have out here is that prairie dog colony that the county told us about, where they were concerned there was some plague activity.
Oh, so all those little dirt hills, those are all prairie dog holes?
Exactly.
Everything looks all dead and boring.
Exactly. This might be our first piece of evidence that there's plague in the area. In fact, this might be a hot zone. All right, come on, let's go get our gear. We'll check it out. So this is a really good prairie dog hole, Brad. It's nice and wide open. Those prairie dogs are probably down deep in there, either dead from the plague or they're hibernating. And what we want to do is go after the fleas. We got to figure out a way to get the fleas to come out.
We've got to call them out?
Well,
All right, I'll give it a try. Come on. Come on. Come on.
Brett, how about we try this?
What is that?
It's a toilet snake.
What is with you researchers and fecal matter?
It's a toilet snake with a clip on the end, and then we can clip on a nice piece of flannel cotton. And that'll mimic the fur of a rodent, and we'll put it down into the hole and the fleas will jump on. But what we really need is actually something on the flannel to attract them to jump on.
Ooh, like cologne? Are the fleas attracted to Calvin Klein too? Or are they more like Old Spice?
Unless your breath smells like Old Spice, because that's what they're attracted to. What it is the carbon dioxide coming out of your breath that makes them think that a rodent is breathing nearby. So we will breathe carbon dioxide in, blow it in the hole, and then see if they'll jump on.
Wait.
We'll shove the snake down in there, see if we can get some fleas to jump on. We'll just snake the flannel down as far as we can, move it around a bit, and then pull it out. And we'll see what we got.
I see fleas.
Oh yeah, there's some fleas on this one. All right, we're gonna put this in a bag and put it on ice.
What's the reason we put them on ice?
Well, we put them on ice so it'll knock out the fleas, and then we can actually study them in the laboratory, see if they have plague.
Are we gonna go check out some more holes so we can have like a larger sample?
We do need more samples. Let's go check out some more prairie dog holes, collect some more fleas, get them back to the lab, and we'll test them for plague. This is what we do as epidemiologists. We go out and try to find clues to why a disease outbreak may have started, try to find some evidence to help us understand how we can maybe stop the disease outbreak.
So you're kind of like a disease detective.
Exactly.
Awesome.
On my next entry into the STEM journals, the biosafety lab and what happened to all the prairie dogs.
Arizona companies see a bright future, from high-tech jobs in aerospace to outer space, renewable energy, crops for the future, and healthcare for the coming decades. Arizona will need more than 150,000 workers in the coming years with skills in science, technology, engineering, and math. CopperPoint supports companies prepared for Arizona's economic growth. CopperPoint.
So did you get some good samples?
I don't know. We got some fleas.
Well, fleas is good. Fleas likely means that we're gonna have some plague in there.
Perfect.
Okay, so Dave asked me if I'd take you through on how to process these. And so to do that, we're gonna grind up the flea and then we're gonna test for the presence of plague.
So just to be clear, you seem pretty excited about this. Are we hoping to find plague or are we hoping not to?
Well, from a scientific standpoint, it's always exciting when we find the disease out in nature. From a public health standpoint, it's better if we don't. So I guess we're hoping not to find plague today.
Perfect. I hope that too.
Okay, so to process these, we have to actually go into a BSL-2. Are you ready for that?
Should I be ready?
I'll show you what to do. You'll be okay.
Oh, man.
Come on in.
What does BSL even stand for?
It's biosafety level, and there's 4 of them, 1 through 4. We're in a 2, which means we need to be cautious, but we don't have to be too worried about what we're working with. And basically all it means is the level of risk that the person working with this stuff has.
So now we're safe?
Almost. We need to put our second pair of gloves on. Can you hand me mine?
2 pairs of gloves?
Yes.
Ready.
First thing we're gonna do is look on the flag that you guys brought from the field to see if we can find fleas.
Should be a bunch of them.
There's quite a few on here. So all we're gonna do is take tweezers and pull off a flea. We stick them in our tube. Now that we have them in the tube, we're gonna use mortar and pestle. Do you know what mortar and pestle are?
I would like to.
All a mortar is just something you squish something against, and a pestle is something you use to squish.
Mortar, pestle. Got it.
So take your tube with your flea, and then take your pestle and squish the flea against the sand that's in the bottom.
And what are we trying to do by squishing the flea?
We're gonna break open the cells and the gut of this flea. So we're gonna release any DNA of the flea, but any DNA as well of the bacteria inside the flea.
Man, this is way more difficult than squishing up fox poop.
Yeah, fleas are pretty tough. That exoskeleton's pretty hard.
And what is the solution that we're mixing it into?
It's called a lysis buffer. And all that means is it is chemicals that help break apart cells. If there's plague there, it releases the plague DNA.
This is kind of like a flea float.
I guess you could call it that.
Because I'm very familiar with fecal floats. I've worked with them before.
Okay, are you done squishing it up?
Ready.
This is actually results from earlier today. So what we're looking at is the fleas we extracted.
Looks like my stock portfolio.
Do you have a stock portfolio?
That's rude.
We ran polymerase chain reaction, or PCR, on the samples. All that is we take the DNA and we go and we look for if there's plague DNA present. And if there is plague DNA, it's actually going to fluoresce. That's what these lines are. And so to start with, we have our positive control. That makes sure everything is working fine in our reaction. That's right here in pink.
Okay.
All of these down here are fleas that were negative, so there's no plague in them. But these two little guys over here, they're plague positives.
Just to be clear, plague positive means that fleas that we found this morning had the plague?
Yep.
I was holding Black Death in my hands this morning.
Mm-hmm.
Ugh. Wait a minute. That's why we didn't find any prairie dogs out there.
That's right. Prairie dogs are highly susceptible to plague. Once it comes into the colony, the whole colony is going to be wiped out within a week.
Oh, poor guys. Now that we found that the plague is present, should we, I don't know, should we call someone? Should we call the Plague Busters?
Close. We'll call the public health department and we'll let them know. They'll go out and they'll do all the preventative measures that are important to keep the public safe.
So I'm guessing if anyone finds themselves around that prairie dog community, they should probably, I don't know, flee the scene.
Still mad at me?
Duh.
Let me make it up to you.
Magic is not gonna work this time, Brad.
No. Not magic.
Yoda's favorite doggy treat.
That's right.
Oh, I want to treat.
I know what he likes.
Oh, you're such a sweetheart.
I know we didn't get along at first, Yoda, but I actually really like this guy.
You are so nice.
Thank you. He's just so...
You want a treat, baby?
He's just so cute and like lovable and...
Good boy.
You just want to pet him and hold him. Aww. Yeah, all the time. I just never want to stop holding him.
We can date again.
Really?
STEM journals, concluding entry. Much like Indiana Jones, disease hunters venture into exotic, sometimes frightening and dangerous settings to learn about stuff that can make us sick. Whether they're gathering data during an outbreak or working with samples in a lab, these people work behind the scenes to keep our communities and us healthy, including that little ice cream thief Yoda.